Hello and welcome back to the Biochemistry for Health Sciences channel. Today we are going to talk about carbohydrates as identification tags. Basically something like your passport numbers or your social security numbers. So previously we talked about the importance of carbohydrates as fuel, especially glucose. And fuel is necessary to provide us with energy. All our cells require energy all the time. So today we'll focus on why carbohydrates are used by cells and proteins as identification. What's so special about carbohydrates that they make such wonderful identity tags? So most of these identity tags, carbohydrates that are used for identification, they usually have usually around less than 10 monosaccharides joined together. And we simply call those small carbohydrates as oligosaccharides. So these carbohydrates can be joined to lipids, and that will give you glycolipids. Carbohydrates can be joined to proteins, that will give you glycoproteins. And then there are basically carbohydrates that have a little bit of proteins attached, and those are called proteoglycans. So here in glycoproteins, the protein is the major part of the glycoprotein. Carbohydrate is a minor component. Proteoglycans is the opposite. Glycans, the carbohydrate is the major component. Protein is the one found in lesser amount. So these carbohydrates are everywhere in the body. Okay, they can be extracellular, outside the cells. They can be part of the plasma membrane. They can be inside the cells, intracellular. Or they can be in the connective tissue, another extracellular form, very, very rich in the connective tissue. So basically these oligosaccharides are everywhere in and outside your cells. So to answer the question, what makes these carbohydrates wonderful identification molecules? The first reason is there are many, many different types of monosaccharides in the body. Here are a few examples: galactose, mannose, fucose, modified carbohydrates, N-acetylglucosamine, N-acetylgalactosamine, N-acetylnurmenic acid, also called sialic acid, glucuronic acid, and so on. Many, many different types of monosaccharides. These two, for example, mannose and fucose, they are very common at the terminals of cells, of microbes, while in humans, the terminals of cells are very rich in this one here, sialic acid or N-acetylnurmenic acid. So you can see microbes have different identities than our cells in the body. So the second reason why carbohydrates make very good identity tags is not only are there a variety of monosaccharides, as we just mentioned, but any two monosaccharides can be attached to one another in many different ways. Okay, so for example, you can attach two glucose, one glucose to another glucose, in 19 different ways. And the reason for that is because of the chemical reaction that attaches two carbohydrates together, two monosaccharides together. So this is basically the reaction. The hemiacetal, so here is a carbon with an OR and an OH, that's a hemiacetal. This hemiacetal reacts with an alcohol of the other glucose, could be any alcohol, this alcohol, this OH, this OH. Alcohol is any carbon that has an OH. Could be this OH, could be this OH. Okay, so basically this can react with any of these alcohols. So there are many different ways of joining these two monosaccharides together. And besides that, the monosaccharides have certain stereochemistry, as we mentioned earlier in the definitions. You can have alpha glucose or you can have beta glucose. You can have alpha galactose or you can have beta galactose. So because of this versatility of this reaction, one hemiacetal can react with many hydroxyls on the other monosaccharides, we can get all kinds of products, and that means we can get all kinds of shapes. And shapes is very important in biology, including identification. Okay, identification. So in biology, we have this very important phenomenon called molecular recognition. How molecules recognize one another. Okay, we'll talk a lot more about this, not only in this video, but in future videos. Very, very important concept that drives the whole biology, the whole human body. Okay, so let's look at an example, hexa 6. You can join 6 monosaccharides together. How many different ways? Billions, 1 times 10 to the power 12. Later we'll see you can make a hexanucleotide, a DNA or an RNA, 4 to the power 6, that's about 4,000 different ways. So you can make 4,000 different hexanucleotides, small DNA or RNA, but you can make billions of monosaccharides, oligosaccharides, billions of hexasaccharides, small carbohydrates that have 6 monosaccharides. For proteins, you can make 20 raised to the power 6, that's 64 million hexapeptides. Okay, so compared to proteins, you can see 64 million, nucleic acids, about 4,000. Carbohydrates, the same hexasaccharide, you can make billions of shapes. So that's why carbohydrates are very good for making all kinds of shapes, and shapes is also our identification. Okay, so the other good thing about carbohydrates is the fact that these are water soluble, and they can be attached to cells or proteins on the outside, and molecular recognition occurs typically on the outside. You know, one cell recognizes another cell, or protein recognizes another protein on the surface. Okay, surface. Okay, and these are water soluble, so we can make all kinds of hexasaccharides or oligosaccharides, all kinds of shapes, many more shapes than we can with proteins, or we can with nucleic acids like DNA or RNA. So basically, these are very rich in information. Shapes is all about information, very rich in information. So it's not surprising we have many proteins that have carbohydrates attached. Okay, these proteins are called glycoproteins. Not all proteins have carbohydrates. There are many proteins that don't have carbohydrates, and then there are proteins that have carbohydrates attached. And these carbohydrates, of course, help the protein in molecular recognition. And that is very important when we talk about how these proteins are transported and then secreted in cells. So where the protein goes after it is made, that all depends on molecular recognition. How stable the protein is, whether the protein is going to be degraded by another protein or whether it's going to be untouched by the other protein, that's also all molecular recognition, and the carbohydrates of the protein will help decide the stability as well as the transport and secretion of the protein. Carbohydrates will also affect the solubility of the protein because carbohydrates themselves are quite soluble, and the carbohydrate may have some effect on the shape of the protein. So it's not surprising that microbes use their proteins. They are called lectins. Lectins are proteins that can recognize carbohydrates, that can bind to carbohydrates. So this molecular recognition between microbes and cells, it's not surprising that the microbes use their lectins to recognize some sort of sugar, some sort of the oligosaccharide that's present on the cell. Here are some examples. E. coli recognizing mannose on your surface of your cells. The type P fimbriae lectin recognizes galabiose, which is another sugar carbohydrate present on our cells, and so on. You can see some examples here. The influenza recognizing this carbohydrate, certain protozoas recognizing galactose carbohydrate, malaria recognizing sialic acid, the fungus recognizing Candida albicans recognizing this carbohydrate. So this molecular recognition is very important. Once the microbe recognizes the carbohydrate, it will bind to it. So molecular recognition will cause the microbe to bind to your cell. And we know from the COVID and many other examples, once the microbe binds to the cell, that triggers all the other events, which eventually leads to infection. So the beginning step of these infections are basically the microbe binding to your cell or to some part of your body by molecular recognition, where carbohydrates play a very, very important role. So there are many examples of molecular recognition that happens in our body. Your immune cells, for example, will recognize microbes as foreign cells and will attack them. That is an example of molecular recognition. Morphogenesis, during embryonic development, how we form tissues and organs, it's all about molecular recognition. When our tissues are regenerated or repaired, involves a lot of molecular recognition. How our immune cells recognize and kill cancer cells is also a lot of molecular recognition. And then there's also cells binding to cells. So during morphogenesis, we want to make sure that all the hepatocytes are bound to hepatocytes and not to some other cell. So there's a lot of molecular recognition that's going on. And interestingly, a lot of this molecular recognition involves oligosaccharides because we can make many different shapes, many different three-dimensional shapes with carbohydrates compared to proteins or nucleic acids. So carbohydrates are not only important for fuel, they're also very important for identification. So keep these two important functions of carbohydrates. One is fuel, and of course fuel means energy, and the other is molecular recognition. Basically, carbohydrates are very important identification molecules, and these two, of course, are very critical for our survival. Okay, let's stop here. We'll hopefully see you in the next video. Goodbye.